O-negative blood is one of the most studied, most debated, and most misunderstood genetic traits in human history. Hospitals stockpile it like a weapon, militaries track it like ammunition, and scientists have spent decades arguing about where it came from and why it still exists at all. The conventional answer is simple. O-negative is a blood type, a rare one. About 6.6% of the global population carries it. It has no A antigens, no B antigens, no Rh factor. That makes it universally compatible. Anyone can receive it. That's why emergency rooms reach for it first. But that explanation only scratches the surface, because when you follow the DNA evidence, when you trace this bloodline through ancient genomes, through plague pits, through Bronze Age collapse sites, through isolated populations on opposite sides of the planet carrying elevated frequencies of the same genetic deletion, the story stops being simple very fast. This is not just a story about blood type, it's a story about survival, about the catastrophes that shaped our species, about the brutal logic of evolution operating over tens of thousands of years, and about what modern science is now doing with what nature spent millennia building. By the end of this video, you'll understand not just what O-negative blood is, but what it means, and why the DNA evidence has forced scientists to completely rethink where this bloodline came from. Let's start with the biology, because you need to understand what makes this blood type so unusual before any of the rest of it makes sense. Picture your immune system as the most paranoid security apparatus ever designed. It checks every molecule that enters your bloodstream. It memorizes threats. It holds grudges across decades. It can distinguish between cells that differ by a single protein. And when it finds something foreign, it does not negotiate. It destroys. Every red blood cell in your body carries molecular signatures on its surface, antigens, biological ID cards that announce exactly who you are to the immune system. Type A blood displays A antigens. Type B displays B markers. Type AB carries both. And layered on top of that system sits the Rh factor, a separate set of proteins that mark your cells as either positive or negative. Most humans carry at least some of these surface markers. They're the flags your immune system uses to distinguish self from foreign invader. O-negative blood carries none of them. No A antigens, no B antigens, no Rh factor. The surface of an O-negative red blood cell is stripped bare, molecularly invisible. Where other blood types wave identification badges at every checkpoint, O-negative moves through the circulatory system like a ghost. The immune system scans it and finds nothing to attack. That invisibility is the source of everything. It's why O-negative can be transfused into any human body on Earth, regardless of blood type, and survive without triggering rejection. Universal compatibility isn't a marketing term. It's the direct biological consequence of a blood cell that has erased its own identity so completely that the body's most sophisticated defense system simply cannot see it coming. Before we start, what part of the world are you in? Drop a comment below, and don't forget to subscribe. Here is where the story stops making sense, at least on the surface. Evolution is brutal and precise. Traits that reduce reproductive success get eliminated, not eventually, quickly, within generations. The genetic mathematics are unforgiving. A mutation that costs you children doesn't survive long enough to become a global phenomenon. It disappears. That's the rule. O-negative broke the rule. And the mechanism that should have destroyed it is one of the most disturbing biological processes in human reproductive history. When an O-negative woman carries an Rh-positive baby, her immune system does something catastrophic. It identifies the baby's Rh-positive blood cells as foreign invaders. It builds antibodies against them, and in subsequent pregnancies, those antibodies cross the placental barrier and begin systematically destroying the unborn child's red blood cells. The medical term is hemolytic disease of the fetus and newborn. The ancient reality was simpler. O-negative mothers watched their children die from what appeared to be an invisible inherited curse. The first pregnancy usually survived. The immune system hadn't fully mobilized yet. But the second pregnancy, the third, the antibodies were primed and waiting. They attacked immediately. Fetal anemia, brain damage, death. Before the development of Rh immunoglobulin in 1968, O-negative mothers faced a hemolytic disease incidence of roughly 1 in 20 Rh-positive pregnancies, with mortality in untreated cases running between 20% and 40%. For tens of thousands of years, this bloodline was paying a catastrophic reproductive price. And yet, here it is, spread across every continent, embedded in populations worldwide, defying every rule. Evolution wrote, "Evolution doesn't make mistakes that last 10,000s of years. If O-negative survived despite killing children, something else was keeping it alive, something powerful enough to compensate for catastrophic reproductive failure." And when researchers finally found the answer, it reframed everything. The mechanism is called balancing selection. It operates when a genetic trait carries both a severe cost and a survival advantage so significant that the two forces reach an uneasy equilibrium. The trait doesn't disappear because the adults who carry it keep surviving long enough to reproduce despite losing children along the way. So what was O-negative protecting against? The infection resistance data is striking. O-negative individuals show statistically significant protection against Plasmodium falciparum, the deadliest strain of malaria, which still kills 400,000 people annually. The reason is structural. Malarial parasites use specific blood group antigens as entry points into red blood cells. O-negative cells, stripped of those antigens, simply offer fewer invasion targets. A matched case control study of Malian children found that blood group O was associated with a 66% reduction in the odds of developing severe malaria. But malaria is only the beginning. Research across multiple pathogen families reveals a consistent pattern. Certain bacteria and viruses use blood group antigens as cellular entry points. O-negative blood, stripped of those markers, forces many pathogens to find alternative, less efficient infection pathways. The evolutionary logic is brutal but coherent. Lose some children to reproductive incompatibility, but ensure the adults survive the pandemics, the plagues, the outbreaks that were killing entire populations before a single child could be born. Nature made a trade, and O-negative was the currency. If O-negative emerged once and spread through migration, the global distribution should follow a predictable pattern. Frequencies highest near the origin point, gradually decreasing with distance, fading toward the edges of human expansion. That's how population genetics works. That's the rule. The actual map breaks it completely. The Basque region of northern Spain carries Rh-negative frequencies between 30 and 35%, the highest concentration on Earth. The Basques are not recent arrivals. Genetic analysis confirms they represent one of Europe's oldest continuous populations, speaking Euskara, a language with no known relatives anywhere on the planet. Their genetic isolation is so complete, they function as living fossils of European prehistory, preserving markers that disappeared elsewhere thousands of years ago. Move to North Africa. Berber tribes of the Atlas Mountains carry Rh-negative rates between 15 and 20% in communities that have resisted outside genetic influence for millennia. Celtic populations of Ireland and Scotland show Rh-negative frequencies of 13 to 18%, rates that appear to predate known migration events into those regions. And then the distribution puzzle deepens further. The RHD gene deletion is now understood to have first appeared in Africa over 100,000 years ago. Its rise to significant frequency in European populations occurred around 30,000 to 35,000 years ago. Multiple independent rises in frequency in isolated populations separated by vast distances and thousands of years of independent evolution. The pattern doesn't resolve neatly, which means the question of why remains stubbornly, fascinatingly open. The distribution map demanded an explanation, and ancient DNA finally provided one. Not a comfortable one, but a precise one. In 2022, researchers analyzing genetic material from Black Death burial sites across England and Denmark published findings in Nature that stopped the scientific community cold. Specific immune gene variants were dramatically overrepresented in survivors compared to victims. The catastrophe hadn't killed randomly, it had filtered. Communities with certain genetic profiles endured at measurably higher rates. The plague was functioning as an evolutionary pressure so intense it shifted population genetics within just a few generations. The Black Death killed between 30% and 60% of Europe's entire population between 1347 and 1351. But the survivors weren't a random sample of who came before. The pattern extends further back. Archaeological sites showing population continuity through the late Bronze Age collapse of around 1200 BCE carry genetic signatures suggesting preferential survival of specific lineages. The 6th century Justinian plague tore through Constantinople and the Byzantine Empire, collapsing populations dramatically. Genetic analysis of survivor communities shows distinct shifts in the frequency of immune-related traits among those who endured. Go deeper still. The Toba supervolcanic eruption 74,000 years ago is believed to have reduced the entire human population to somewhere between 3,000 and 10,000 individuals. Genetic modeling of Toba survivors shows patterns consistent with strong selection for infection-resistance traits. Every major catastrophe in human history functioned as a filter. Each one concentrated certain survival profiles in the populations that remained. This isn't mutation persistence. This is a species being repeatedly forged by extinction. On the morning of September 11, 2001, as the first tower fell, New York trauma surgeons faced an impossible situation. Hundreds of casualties arrived simultaneously, blood types unknown, no time for compatibility testing. There was only one answer: O negative, always O negative. That scene has repeated itself across every major disaster in modern history, and each time the same shortage crisis unfolds with terrifying speed. Every 2 seconds somewhere in the world, O negative blood flows into a stranger's veins. Car accidents at midnight when blood banks are closed, mass casualty events where victims arrive faster than lab results, combat zones where soldiers bleed out before medics can run a single compatibility test. Despite representing less than 7% of donors, O negative provides the entire universal emergency blood supply. Every level 1 trauma center on Earth depends on this microscopic minority of genetic outliers. The shortage of mathematics is brutal. O negative donors can give to anyone, but they can only receive from other O negative donors. Universal givers locked inside a genetic prison of scarcity. During Hurricane Katrina, O negative stocks dropped to 12-hour supplies. The 2010 Haiti earthquake triggered global shortages lasting months. The Las Vegas shooting in 2017 depleted the city's entire O negative reserve within 6 hours, triggering emergency transfers from Los Angeles, Phoenix, and Denver simultaneously. Military planners track O negative supplies the way they track ammunition. Forward operating bases without adequate O negative stocks couldn't handle mass casualty events. They had to evacuate critical patients to bases that did. The curse that nearly erased this bloodline across millennia had become the most strategically valuable biological resource in modern warfare and medicine. For tens of thousands of years, O negative carried its reproductive curse without relief. Mothers faced hemolytic disease in pregnancy after pregnancy. The bloodline survived only because the adults carrying it kept outliving the catastrophes that killed everyone else. That brutal balance held until 1968, when Rh immunoglobulin eliminated the incompatibility problem entirely. For the first time in human history, O negative carried no penalty, only advantages. And science didn't stop there. Researchers have since demonstrated that CRISPR gene editing can be used to engineer O-type RHD negative blood cells in a laboratory setting. Not discovered, not extracted, built. Scientists reverse-engineered what evolution had spent tens of thousands of years constructing and produced it synthetically. The molecular invisibility that O-negative blood achieved through catastrophe and selection pressure can now be copied, replicated, and scaled. The implications cut in two directions simultaneously. On one side, engineered universal donor blood could end the shortage crisis permanently. No more depleted reserves after mass casualty events. No more emergency transfers between cities after a single disaster. No more strategic vulnerability in combat zones. On the other hand, the fact that science moved so urgently to replicate this specific trait raises a question that researchers don't answer directly, but that the data keeps circling back to. Why does humanity keep working so hard to preserve and now mass-produce its most ancient pandemic defense? What does evolution know that we are only beginning to understand? What threat, known or quietly anticipated, keeps pushing science toward the same solution nature reached so many thousands of years ago? The answer may already be emerging, and O-negative blood may be the only thing standing between us and it. O-negative blood is not a glitch. It is not a curiosity. It is not simply a rare trait that happened to survive. It is a record, written in human DNA across tens of thousands of years of catastrophe, survival, and adaptation. Every population that carries it in elevated frequencies tells the same underlying story. Something came for our ancestors, something deadly enough that nature was willing to sacrifice children to keep adults alive long enough to outlast it. And outlast it they did. The biology is layered. The immune invisibility is real. The reproductive cost was real. The infection resistance that balanced that cost was real. The ancient DNA from plague pits and collapse sites and volcanic bottlenecks is real. And the laboratories now engineering what evolution spent millennia building are real. What the full picture tells us is this: O-negative blood is not the blood of aliens or lost civilizations or conspiracies. It is the blood of survivors. Ordinary people who faced extraordinary threats and carried a genetic advantage they never knew they had. Their descendants walk among us now, still carrying that ancient code, still donating it to strangers in emergency rooms at 2 in the morning. If you carry O-negative, consider donating. If you don't, consider donating anyway, because this story isn't really about a blood type, it's about what humanity has already survived and what it might need to survive next.